Okay, this video is all about feedback and positive and negative feedback loops and mechanisms. So, what we mean by feedback is just a signal within a system that's used to control that system. So, something happens, and then in response, something else happens to help control what's going on. That's feedback essentially. Now, a feedback loop is when you have the feedback occur, a response happens, that's considered to be a feedback loop, right? So with this little diagram here, right, A happens, A causes B to happen. B then can have an effect on A. That's what we call a feedback loop. So this is found in many living and non-living systems. It's found in things that are engineered. It's found all over the place, but it is found, like I said, in nature. Now, you can either have positive feedback. And what positive feedback basically means is that when A happens and causes B to happen, B then actually reinforces and increases the amount of whatever A is. So it's a positive feedback loop in that when A happens, B causes A to happen even stronger or even more or even faster. That's a positive feedback loop. Now, a negative feedback loop does the opposite. Negative feedback is when something happens with A that causes B to happen, and B kind of counteracts or mitigates whatever's happening with A. So positive feedback loops help accelerate some sort of change. Negative feedback loops help counteract or inhibit change. And so negative feedback loops are really important for maintaining homeostasis. So like, for example, right, you start to get hot. What happens? You start to sweat. That sweat helps cool you off so you're not as hot anymore. That's negative feedback. That's homeostasis, right? Maintaining body temperature. So we're going to look at some examples here today. All right, so like I was just talking about actually with body temperature, that is our first example here. And here's a diagram of the negative feedback loops that are happening here, right? So normal human body temperature, right around 98.6 degrees Fahrenheit. Of course, this varies from person to person a little bit. You might be a little bit above that, might be a little bit below that. But if the body's too warm, then a variety of things happen, right? So let's take a look, right? Body temperature rises, then a bunch of different things happen. Blood vessels dilate. That helps bring blood to the surface of your skin so that it can radiate heat away. Sweat glands secrete fluid so that you can take advantage of the heat of vaporization of water to draw heat energy out of the skin again. Those two things, of course, are going to help you lose heat to the environment to bring you back to a normal body temperature. Now, with most any of these homeostasis kind of loops, right, we're trying to stay at a set point. So usually there's going to be a kind of mirrored image like you see here, where there's actually two negative feedback loops that work together to keep something at a certain point, right? Because you could also have your temperature get too low, right? Body temperature could fall. In that case, blood vessels constrict so the heat is conserved, right? So your blood stays more in your core. If you go outside on a day like today when it's really cold, your more of your blood's going to stay with your core internal organs and recede from like your fingers and toes. And that's why people get frostbite and they can lose like fingers and toes, because the blood literally stays back in order to try to keep your body temperature up. Sweat glands don't sweat, and shivering starts to happen. So your muscles kind of involuntarily activate in order to generate heat. And the whole point of that is to bring that body temperature back up, right? So we have these two loops, right? These two feedback loops are both negative feedback loops that help maintain that consistent temperature, right? And so that's an example of negative feedback. Positive feedback, one classic example of this is blood clotting. So again, as a reminder, positive feedback is where a change happens, and then that causes something else to accelerate whatever that change is. And so in blood clotting, when platelets get activated due to like the release of a wound, they actually, upon activation, release chemicals that activate more platelets. So you cut yourself, there are chemicals that get released, signaling molecules, little hormones essentially, that are released from the wound site that attract and activate platelets, thrombocytes, right, our fancy word for platelets. Those platelets then travel to the site, they get activated, they start to form a clot, and every time a platelet gets activated, it releases more of those chemicals. So those same chemicals are causing additional platelet activation. So this accelerates the process so that you can quickly clot the wound, right? So you don't wind up continuing bleed and bleed and bleed and bleed, right? We want this to happen quickly. And so positive feedback loop like this, right? This is the loop right here, helps us clot that wound up quickly. Another classic example is childbirth. So pick wherever you want. You can start anywhere on this cycle here, but we'll start here, right? So the head of the baby pushes against the cervix, okay? So you have the cervix right here. When the baby's head pushes against here, that causes nerve signals to be sent up to the brain of the mother, and the brain then stimulates the pituitary gland to release oxytocin. And oxytocin is another hormone, so it's a chemical messenger molecule, right? And that hormone oxytocin, actually, once in the bloodstream, it goes to the uterus and it actually causes the uterus to contract. So that oxytocin stimulates uterine contractions, and of course, as the uterus contracts, it pushes the baby's head against the cervix. And then the cervix sends nerve signals to the brain, and so on and so forth, right? And so you keep getting more nerve signals from the cervix, more oxytocin in the bloodstream, stronger contractions. More nerve signals, more oxytocin, stronger contractions, more frequent contractions, and eventually get to the point where, full-blown labor, right? And she's pushing, and eventually the baby comes out. That's another example of a positive feedback loop, right? So you can see how all these things eventually just build on each other, right? So positive feedback is all about accelerating some change, adding to it, whatever it is. Negative feedback is all about counteracting or inhibiting changes. So what you're going to be doing in your lab journal, although if you wanted to do it digitally and submit it digitally, you could do that if you want, is you're going to research and diagram glucose feedback loops. So how does the body maintain normal blood glucose levels, right? So you're going to need to research the action of both insulin and glucagon. We've already talked about insulin. You should be pretty familiar with what insulin does. Insulin is that hormone secreted by the beta cells of the pancreas, right, that help bring blood sugar levels down. Right, so you're going to look into how that works. So you should already know. We've already covered it, right? Glucagon does the opposite. So just like with body temperature going up or body temperature going down, right? Your blood sugar levels could go up when you eat. Your blood sugar levels could go down, like especially if you're exercising, doing some sort of strenuous activity, right? So if there's a drop in blood sugar, we need to bring that back up, right? The cells of the body need to make sure that they've got access to that glucose. And so you're going to research what glucagon does. And so here is kind of an overall map of how you could do this, right? So just like with the body temperature one, right? You got your normal levels, and then a change happens, right? So these little pink arrows here are representing a change. Either blood glucose level rises or blood glucose level drops. What happens next? So the thing that's going to happen next, right, is that the pancreas is going to do something. And it's the pancreas in both of these situations. Some other things that are going to be involved in addition to the pancreas. So some other terms you should have in your diagram. You should include, of course, insulin and glucagon. You should talk about the beta cells of the pancreas. You should talk about the alpha cells of the pancreas. And you should also talk about, of course, glucose, but also glycogen. And glucagon and glycogen look very similar, but they are different words. They are pronounced differently, and they are very different things, but they have an impact on each other. Unfortunately, all these words kind of sound the same, and that's a little bit tricky, right? It's because they all do stuff with glucose. We got glucose, glycogen, glucagon. Different words mean different things. So when you have this feedback loop done in your lab journal, make sure that you look at the instructions on MyPLTW2. There's a couple other things going to have you add. It's going to have you note what goes wrong on type 1 versus type 2 diabetes on your diagram. Come up, show that to me. I'll grade that for you, and you'll be good to go on to the next thing.